Our next speaker, I don't know why I just changed positions. Our next speaker is Professor Jessica Green. She's a professor of biology at the University of Oregon, and she's also the Chief Technical Officer of Phylogen Incorporated. Jessica?
Are we not all supposed to be sitting up there? Just the leader of the group. I just thought we were supposed to be there.
We didn't want to tell you.
You didn't want to tell me, yes. Okay, oops. So Shelley, I can answer that question right out of the gate. If I were to do this again, the answer would be yes. All right. Okay, super. And why does my clock say 4 minutes right now? Did that all take 4 minutes?
We're back.
Okay, I want to get this right.
You're back to 15.
Great. Well, I want to start off by acknowledging, Let's see. This is exactly the same thing you used, right, Jonathan? But maybe, I don't know. It is? Well, I think you have a copy of the talk back there as well, if this doesn't work. I just figured what happened after Susan's talk. Hey, look at that. Great. All right. So I wanted to start off by acknowledging all of the great people that I've had the pleasure of working with at the Biology and Built Environment Center and our funding sources and collaborators. Our current director of the Biology and Built Environment Center is here, Kevin van den Wijmelenberg. Can you raise your hand, Kevin? And please take an opportunity to talk to him at this meeting. I also, Erica Hartman is another key player at the Biology and Built Environment Center, a current co-PI, and she is also here. Erica, can you raise your hand? Super. And so thanks to all of these great people, and particularly Brendan Bohannon and Charlie Brown, who both played a seminal role in founding the Biology and Built Environment Center. I'm going to give you a brief history of BioBE Center research, which started with the founding of the center. And we've done many different projects. I'm not going to talk about them all today, but we have done some work. Oh, so my slides are not advancing. Oh, it's because I'm doing it on my laptop. You guys need to fill me in on, there you go. That's all of the people that I'm acknowledging. And this is a timeline that I'm going to be reviewing. So, some of our earliest work was focused on ventilation and how it impacted the hospital microbiome. We've done some work looking at how building design and operations influenced the classroom and office microbiome. Later on, we looked at how the microbiome of classroom surfaces were mediated by human contact. We've looked at the human microbial cloud and its contribution to the indoor environment. We've done some work looking at how urban green space impacts the outdoor air microbiome, and finally some work looking at antimicrobials and antibiotic-resistant genes in dust. And I'm going to talk about a small subset of these projects today, or these publications. We're currently at the Biology and Built Environment Center at the University of Oregon, that's where we're located, looking at the effects of light, sunlight, on the dust microbiome in terms of viability and composition. We're looking at, through an EPA grant, home weatherization and how that impacts indoor microbial diversity, the health of occupants, and how that interacts with their behavior. We're again doing more work looking at antimicrobials and antibiotic resistance in athletic facilities. And we have a great postdoc, Ashkan Fahimipur, who's looking at theoretical ecology models to understand indoor microbiome dynamics. We're doing some work looking at microbial transmission from plants and dogs to the human skin and looking at how that's mediated by the indoor environment. And finally, we just got a grant that's co-sponsored by it's Tallwood and the USDA, looking at the influence of building materials on indoor microbiome exposure. I'm going to tell you a few data stories that are kind of in these three different narratives, one looking at ventilation, daylighting, and antimicrobials on the indoor environment, and I'm going to start with ventilation. So this is historic at the Biology and Built Environment Center. The first question that we addressed is how does ventilation impact the indoor microbiome? It talk about the history of this field and indoor environmental quality. It's known that ventilation impacts indoor air quality from many different aspects, particularly physics and chemistry, and we wanted to use molecular biology to understand how it impacts indoor microbial ecology. The first project that we worked on was in the hospital environment, where we asked a simple question: how does ventilation, and namely window ventilation versus mechanical ventilation, impact indoor bioaerosols? And what we found is that if you take these air samples, put them in a statistical map in ordination, and first look at outdoor air samples, which are in the green, and then compare those to the community composition in mechanically ventilated rooms, which is blue, you see very little overlap in the types of bacteria that are in mechanically ventilated rooms. And what we found was that mechanically ventilated rooms were dominated by the people and the skin microbiome, oral microbiome, gut microbiome that were occupying these spaces, relative to the outdoor air samples, which primarily had microbes that you find in plants and in dirt. We also looked at bioaerosols in a classroom setting. Charlie Brown, co-founder of the Biology and Built Environment Center, designed the classrooms, the ventilation systems in these classrooms to have something that's called a night flush, where you let outdoor air into the rooms through louvers, and that turns over the air in these spaces. What we found is that if you look at these, first look at the very top panel, you're looking at outdoor air and how outdoor air bacterial community composition changes through time. We did this study over 2 weeks. We selected a time period where you had a significant shift in the outdoor bacterial community composition. And if you look at the second panel, you'll see that these night flushed rooms, even though there's a time lag, they pick up on this outdoor turnover of bacteria, and you see those bacteria showing up in the indoor rooms, whereas you see a much more dampened effect in rooms that did not have this energy-efficient night flush ventilation treatment. And finally, in the business school at the University of Oregon, we looked at the dust of faculty. The faculty members could choose offices that were either mechanically ventilated or window ventilated. And what we found, again, this is a statistical map. Every sample is a dust, or every dot's a dust sample, and dots that are closer together have more similar bacterial community compositions. You have a really different profile in the mechanically ventilated rooms versus the window ventilated rooms. And if you do a deeper dive and look at specific taxa, you'll see that taxa like Deinococcus, for example, are more prevalent in the mechanically ventilated rooms, and methylobacteria is more prevalent in the window ventilated rooms. So if you were to, I think, ask the majority of the people in this field whether or not ventilation impacts the indoor microbiome, the answer would be yes. And the next step is to understand what is the implications of this. We're doing some work looking at daylighting as well. Kevin van den Wijmelberg is a world-renowned expert in indoor daylighting. Sunlight is a very important attribute of architecture and design, and so because of this, we wanted to know, how does daylighting and sunlight impact the indoor microbiome? And again, there have been some studies using culture-based approaches to show that daylighting can impact, for a few species, the viability of microbes indoors, and we wanted to look at this using molecular tools to know if it was a more general pattern. So what we did is, this work is being led by Ashkan and again Kevin, there's many people contributing to it, is we use experimental microcosms that are basically like little built environments where we wanted to know if you put dust into these microcosms and expose them to different types of light, how that impacts the microbiome. And so what we did was we had 3 different types of glazing where we essentially got a gradient of dust that was exposed to visible light, UV light, and then some that were completely in the dark. And this was Oregon dust, so a lot of this work is really based on the microbiome that you find in the Pacific Northwest, and we left these dust samples exposed to light for 90 days. And we also, inspired by a working group, I believe, that was led by MicrobeNet on understanding the viability of the indoor microbiome, we used a method, it's probably not perfect, but it's PMA to characterize the viable microbiome in these samples. And what PMA does is it penetrates dead cells and it inhibits PCR amplification of those cells. And we also used real-time qPCR so that we could understand not just the relative abundance of microbes, but their absolute abundance. And the first thing that we wanted to look at is, does light exposure impact the viability of bacteria and dust? So these are violin plots, and what you're looking at on the y-axis is a measure from our qPCR data of the total number of 16S gene copies on a log scale in these samples. The first thing that I want you to do is just look within a treatment. So again, the gray and black is the dark dust. The red is the visible dust, and the blue is the dust that was exposed to UV light. If you look at the dark dust, you'll see that there is a significant proportion of the community that's living, and that's something that has been a matter of debate in this field, is whether or not microbes are alive in the indoor environment. So it's about 10% of the dark dust was viable, and in the dust exposed to light, it was around 5%. And then the second thing is, if you look across these treatments, take the viable portion, for example, the dark, going from dark to the light, and if you look at the viable proportion, you'll see that it decreases going from dark to light. So light exposure does seem to reduce the viability of bacteria and dust. Now let's look at community composition. If you look at the total community, which you're highlighted here on the right, again using an ordination map, you'll see that the composition of dark dust is fundamentally different from the composition of dust that has been exposed to sunlight. And the size of these, every dot is a dust sample, and the size of the dot tells you something about the total bacterial abundance in that sample. And we see the same thing if you look at just the living proportion of the community. You see that you have a different microbial community in samples that have not been exposed to light relative to ones that have been exposed to light. Now, if you want to understand more and kind of do a deeper dive about the composition of this dust, what we're looking at here is, again, violin plots where we're looking at on a log scale, the proportion of genes that are associated or commonly found in outdoor air versus human skin. So the way to read these plots is, if you're in the area of the violin towards the bottom, you have genes that are more commonly found on human skin, and if you're on the top, you have genes that are more commonly found in the outdoor environment. And if you, again, look within a treatment, so let's look at the dark dust, for example, you'll see that the viable part of the community has more outdoor environment-associated microbes, and the dead part, which it's total, it's dead, dormant, and living, has microbes that are more commonly associated with human skin. And then if you look across treatments, if you look at the viable portion, you'll see that as we go from having no exposure to light to having exposure to light, you get a community that is more dominated by microbes that you would commonly see in the outdoor environment. So if you were to ask me, does daylighting influence the indoor microbiome, I would say yes, based on what we know from culture-based studies and also from this experimental microcosm, but of course there's much more work that needs to be done in this arena. The last data story that I want to talk about is our work with antimicrobials. And we wanted to know, does the concentration and diversity of antimicrobials in the indoor environment, does that correlate with the indoor microbiome? And so I want to clarify and share with everybody one of the reasons that we were really interested in looking at this relationship between antimicrobials and antibiotic resistance genes was inspired from working with Erica Hartman, who did a lot of this work with antimicrobials as a graduate student before joining our group. And so what Erica pointed out to us is basically when you think about what's going on in dust, we know that dust is filled with skin, it's filled with insect parts. We had been very concentrated at the BioBE Center and focused on looking at the microbiological aspects of dust, including bacteria and fungi. We had done less work with viruses ourselves, but what Erica pointed out to us was that this dust is laden with chemicals, and these chemicals matter for indoor environmental quality. Chemicals like antimicrobials, chemicals like plasticizers, and flame retardants. And so what she was interested in pursuing was understanding all of the different attributes of what's impacting indoor environmental quality. So not just design choices like ventilation and daylighting, but also human behavior and the antimicrobials that humans are bringing into the indoor environment. Now, the antimicrobials are pervasive in the built environment. Again, we bring them in for our personal care products. We bring them in our cleaning products. You can buy paint that is antimicrobial, and of course you can get countertops and chairs and floors that have antimicrobial materials in them. The first study that we did was in collaboration with Curtis Huttenhauer and Rolf Halden to understand, and many other investigators, to understand the relationship between antimicrobials and antibiotic resistance genes in dust. This work was published in ES&T, and we looked at a historic building at the University of Oregon, which again, we're looking at the total abundance of antibiotic resistance genes in these dust samples from this historic building. And if you look at the pink violin plot, that's our building, Gerlinger Hall, which has a relatively small amount of antibiotic resistance genes compared to what's been published in other studies on the built environment, and also compared to studies that have been reported for the human gut. And that's because Gerlinger Hall is a historic building. It's a leaky building. This is a photo of one of the windows of this building from the inside of the building. That's how leaky the building is. And so we knew that if we found any pattern here, that would be a very strong signal given how few antimicrobial resistance genes were in this environment. We did find that there was an association between the abundance of certain antibiotic resistance genes and the concentration of certain antimicrobials. So an example would be the relationship between ERM-X and triclosan. We found a significant association between these two. A lot of these genes are resistant to many different types of antibiotics, and the Z-Pak being one of them. And this is an example of looking at the relationship between triclosan and one of these antibiotic resistance genes that we found. We've now moved on to try to understand, is this repeatable across many different buildings, not just one building? And so the team has analyzed dust and collected dust from dozens of gyms across the Pacific Northwest. And this is a teaser of the data. We're still working our way through it, but this is a plot that is really common in ecology, where we're looking at the dissimilarity of bacterial community composition as a function of the dissimilarity in the chemical composition. These types of plots you see them a lot in soil microbial ecology studies, and an ecologist would interpret this pattern to say the environment is selecting certain types of bacteria into these samples, and in this case it's the chemical environment. Now if you do a deeper dive and look at if you have an antibiotic resistant gene that is present in a dust sample, is it correlated with the concentration of an antimicrobial in the sample? And yes, you can see here for vancomycin resistant genes, this is one example of how it's correlated with ethylparaben. And some of the modeling that we're doing is to really understand the dispersal of microbes at the organismal level, and then what might happen once it lands into a particular environment, how it may be interacting with the chemistry in that environment. So to answer, do antimicrobials influence the indoor microbiome, I would say stay tuned. And in closing, I just wanted to remind you of these 3 areas, data stories that I shared with you today, and share with you again sort of the founding of the Biology and Built Environment Center, which again was with Charlie Brown. When I first met with Charlie, he said, I've dedicated my career to understanding the impact of architecture on 2 attributes of building performance. One of those is how this building is impacting the outdoor environment. So is my building contributing to global climate change? Is my building contributing pollution? And the second was really about human experience in that indoor environment, to understand how do people feel when they're in a building that I've designed. And we've now added this 3rd dimension to our analyses, which I think of as the 3 legs on the stool, to understand how does my building impact indoor environmental quality with this new microbiological lens. Thank you.
We won't have any time for questions, but I'm sure you can catch up with Jessica in one of the break periods.